So the last speaker before the coffee break is John Donoghue. He's a professor of neuroscience at Brown University, and he has been working on neural coding in the context of how we generate motor actions. And when Al and I were talking about people to invite for the meeting, one of the things we were looking for in speakers is people who sort of sit at the intersection of technology, basic science, and in some cases applications. And I think John's a great example of that. Aside from generating a lot of basic insights into how populations of neurons and their activities lead to motor output, he's really been at the forefront of developing new recording technologies to look at recording of large populations and also at the application of these technologies for what we hope will be therapeutics. And he has some really exciting work looking at neural prosthetics and how you can take these codes and generate actual motor actions in patients. So without further ado, John.
Thank you. Thank you. Good morning. Can I have the slides up, please? Actually. Good. So my lab is involved, has sort of two parts, artificially divided. One is very basic, interested in how you turn the computations that underlie the transformation of plans into actions in cerebral cortical networks. And because of that work, about 15 years ago, some very applied work emerged, and particularly this work on developing brain-computer interfaces, devices to help people who are paralyzed now reconnect their brain to the outside world using these insights that we've gained from our basic work in how plans become actions. And our project is called BrainGate. So what I thought I would do is I would use the work we do in brain-computer interfaces as a way to illustrate excuse me, open questions in brain function. And so I'll take you at a very or reasonably high level through the work that we've done, and then I hope lead you back to ways of or discussions about how the advances in this field will depend on a greater understanding of how the cortex actually does compute plans into action. So let me start with talking about Kathy here. Kathy is one of the BrainGate participants. She's one of 5 million people who are paralyzed. She, in fact, has very severe paralysis. She had a brainstem stroke 15 years ago. She's unable to volitionally use her arms or legs or any part of her body, and she can't speak. But I'm going to start here with the end. This is where we are today. This is Kathy controlling this robotic arm. Now, Kathy has, every morning she has someone deliver her a cinnamon latte. It's what she has as her morning coffee. Of course, she's completely reliant on other people to do anything, like brush her teeth or have a drink of coffee. But here, she's using our device. She has a tiny, it's a chip, it's a 100 microelectrode array implanted in her motor cortex. It's coming out through a plug in her head. It's being decoded and used to run this rather fancy robotic arm. And she's completely controlling the actions of that robot with her thoughts. She's imagining motion. And she's having herself serving her coffee, or serving herself her morning coffee, for the first time in 15 years. And this was actually the second attempt at doing this. So again, I want to use this as a way of explaining how important it is to understand how the brain works, so we can take and actually produce devices like this. So what I want to do is take you through the series of problems we had to solve as a group to try and come up with a device like this. And I think the reaction to the device is I think, in this kind of iconic image of her, which appeared all over the place when we published this work, of how important it is to regain even this very small amount of independence. So the goal, basically, of brain-computer interfaces for us, or BrainGate, is basically to give you back your arm. And we want to have something that's safe, and usually our own arms are safe, useful, natural to use, it's reliable, and of course as portable as our own arm. These are very ambitious goals, and you know, as you see, we aren't there yet. So the challenges really are to identify, to build this bridge between the brain and the outside world, are to understand where do we get the signals, what kind of signals would we use, what are the sensors that could give us those signals, how would we decode them, and in the clinical setting, what are the assistive technologies, what are the devices that would help these people. And this really is requires a consortium of neurobiology, computational neuroscience, neuroengineering, and clinical folks to all work together to try and tackle each of these questions. So I'm going to walk you through how we thought through these questions, and you can see the adequacies or the inadequacies of the answers we came up with. So when you think about reaching and grasping or using your arm, there's actually, we now know, there's probably a dozen or more areas involved in a very highly interconnected network that is involved in generating arm actions. And so all of these sites are potential sources of signals that could be command signals for the arm. But among this whole collection of areas in premotor areas and parietal areas and in the primary motor cortex, the primary motor cortex itself is really a desirable target, actually for some practical reasons, like we know where it is in humans and monkeys. We actually have a huge amount of data on arm control in monkeys based on years and years of electrophysiology. And as best we know, it has the closest direct link to movement. It's kind of a conduit that the other areas focus on, even though it is embedded in this network. So we've chosen the primary motor cortex as being pragmatically oriented as the site. Now, in terms of what kind of signals would you use to control such a device, so we've known since the 1930s that extracellularly you can record two types of signals. One's called a field potential, the other's called an action potential. The action potentials are really known as the code based on the work beginning with Adrian. But there is this other signal, these field potentials, which can be recorded outside the brain or inside the brain, that represent another kind of signal. And I'm very pleased that, in fact, the brain-computer interface world has taken or forced the field to really take a much closer look at these two signals and their utility as a possible command source. As many of you know, that the field potentials have really gone in a direction of clinical use in clinical applications like sleep and epilepsy, and have kind of fallen by the wayside in sort of fundamental neuroscience until recent years. And interestingly, this just video, which is actually from a single electrode in a human, points out how different these two signals look. So even not knowing much about what's going on here, you can see that there's actually quite striking differences between the signals that can be pulled out of even a single site. But for the time being, we've focused very heavily on action potentials. We've known since the 60s that these action potentials in motor cortex, their patterns contain information about movement. And from the work of Georgopoulos and his colleagues since the 80s, we know that populations of cells encode information about hand motion through space and probably lots of other details that I'll show you about. So basically, we realized that because these are populations of neurons that are encoding these patterns, that motor commands emanate from spiking patterns. And I'll use this QR code as kind of an emblem of the patterns of activity that come out of a sort of patch of cortex with sort of, think of each little black and white spot as a neuron reporting a neuron's activity. And of course, in a particular field of cortex, this is flickering 100 to maybe even 1,000 times a second. So in order to get ensemble codes, you have to have electrodes that can record lots of neurons. And I think we're at a point where we can get some of that, but we're not anywhere near what we need to do. So we had to develop a sensor. Dick Norman developed this sensor in the beginning. It's 100 microelectrodes that can be fabricated in a little rigid array. But in many regards, it's not a whole lot unlike what Adrian used in the 1930s as a classic microelectrode. They're in a 10 by 10 matrix. They can be implanted in the brain and in their current configuration made for either animal work or for humans. It consists of an array that's implanted in the brain and a plug on the head, and that's what that thing was on Kathy's head, was where we connect the plug up to get to the outside world. So we developed that sensor, and with it, we tested it first in animals. And I won't go into any of the decoding work, but of course that's a major aspect of this, is when you get a pattern of activity out of the brain, can you make any sense of it? And I think this is a major field of looking at collections of neurons and trying to understand what is the information contained in now small collections of neurons. So we know we can get things like from two different patterns, we can distinguish intended direction, like moving your hand to the left or to the right or up or down or forward or away. And by decoding these patterns, we can extract these in relatively rapid time intervals. So they're on my website and in these papers, you can see lots of descriptions of the decoding, but I won't go through those. So in 2004, actually a rather complex path, we put together a clinical trial to test in humans the safety and goals of trying to understand these brain-computer interfaces. And we used this array we developed. It was implanted in the arm area of motor cortex in humans who were paralyzed, severely paralyzed with a plug in their head. They generated patterns of activity, and the idea was to decode these through this cart full of electronics that's basically your lab in a small box. And it has to do real-time data processing. So for the patient, they think about moving a cursor, and then the cursor moves on the screen so they can connect their brain to the outside world. So far, we've implanted 7 people. All of them are tetraplegic. Some have ALS, some with spinal cord injury, some with stroke. I want to start by actually just showing you the activity of one single neuron and listening into that activity, because now you're actually hearing the motor cortex of a human. And I want to point out a couple of things after you listen to the activity. So there'll be a technician giving verbal instructions to this person to open and close their hand, and I think you can see quite easily and hear the rate code that's going on as they think about, now they can't move, but they're thinking about moving. If we could have a little bit more volume, please.
Relax. Close your hand. Relax. Open your hand.
So I think it's quite easy to see here that, in fact, merely thinking about action is activating the neurons. This has a number of actually very interesting implications for human neuroscience and human disease. One is, this is the primary motor cortex that's supposed to direct movement, and in fact, this person immediately activates this neuron upon thinking about moving when they actually can't move. In addition, this person at this time had been paralyzed for 12 years. They hadn't used their motor cortex to do anything, and yet it was still active many years after paralysis. And it was active almost all the neurons were selective for hand action. And so we are perplexed by where is plasticity? This is a part of the brain that wasn't used to do anything as best we know, and yet it in fact didn't show any plasticity. So these are actually important lessons that we got instantly as soon as we turned on the amplifier. We were actually looking at the human brain at a level, here only one neuron, but now at the ensemble level, and really gaining insight into both how it works and also how it can be harnessed to control devices. Now, in the early days, I'll let you listen in on Matt controlling a cursor.
Okay, so here's the Cyberkinetics desktop. What are you going to do first?
Open my email first.
Okay.
So he can simply open icons
You can open the first one.
by moving this arrow or this cursor over an envelope or something.
Congratulations, you were doing a great job.
Very good.
So this was our early 2006 very simple decoder kind of control, this wobbly cursor. Over the next several years, we worked with computational neuroscientist Michael Black and his colleagues in particular, and we tried to extract as much information as we could out of this signal that was coming out, and we got much better cursor control. Here's the cursor going from spot to spot. There's a target, and in fact, we showed that by imagining squeezing the hand, you could click and create a click signal so that you could do actual point-and-click movements. But in fact, if you look at these, they're not as smooth or natural as our own mouse movements. They're pretty accurate. In fact, it would give people enough time, they can actually move to an icon on a screen or a dot, but they're slow. Just getting the cursor to the target is about twice as long as an able-bodied human, and if you have to futz around and get to the small target and click, some of these trials, they'll take on the average of 9 seconds. So they're much slower than we are, even though they have a reasonable degree of accuracy. But even with cursor control, you can ask the question, what can you do? And these are projects that we're developing with collaborators largely to say, what can you do with this kind of technology? So one, with a point-and-click action, you can do one-finger typing, and we have patients who are communicating. We actually have one GChat with us. As I showed you in the beginning, you could have a device, and this is something we're aiming towards, where there's a fully implantable array, some kind of iPhone-like device that transmits to a home device that will then give you your drink of water whenever you want. As you saw Kathy, she had her morning coffee. In fact, you could apply the same approach to prosthetic limbs for people with limb loss. You could now use the same system and control advanced robotic limbs, which are actually already here, so that amputees could once again control their arm in a way that's like their own natural arm. And one of the major long-term goals that we have would be to actually transmit this system to an implanted, wirelessly communicating electrical stimulation device that would drive their own muscles. And so this part is actually not so far-fetched. In fact, it's this FES system. There are about 600 people that have an externally switch-controlled FES system that can move their partially paralyzed arms. So that kind of system isn't completely beyond all science or science fiction. So this actually, getting to this point of having some control, led us back to the basic monkey lab kind of question of saying, well, actually, how much control can you get out of a small patch of cortex with these electrode arrays? And many of you remember that from this Penfield map, which Penfield himself admitted was not an accurate map, that the arm is actually spread out over a large area of the motor cortex, when in fact it's actually not true. In fact, there's probably no evidence to support exactly this map at all. And in fact, and what I'll show you is that really there's a huge amount of information packed into small volumes of motor cortex. So this work in human drove us back to the lab, and we, thanks to Carlos Vargas Irwin and Michael Black, we set up a lab in which we could have monkeys catch objects swinging on a string, as you see here. We use motion capture techniques, and this is the actual arm movement of the monkey catching this object. And then we asked the question, based on the activity we recorded, how much could we actually detect and decode from the motor cortex? And I'll just show you this movie that summarizes that experiment, where the ghosted image here is what the monkey was actually doing, and the solid image is what we were able to reconstruct in real time, or reconstruct from the neural activity in this small patch of motor cortex. And although clearly it's not perfectly accurate, we can decode the entire limb, reach, grasp, hand motions, wrist motions, all from this single collection of cells, which bodes very well for being able to recreate that motion from an electrode array implanted in motor cortex. So we did that, and you've already seen the big payoff. Here's Kathy in a setting using one robotic arm. We had her drink her morning coffee, as I showed, but we also did something a little more quantitative. And here she is using, actually, this is one of these prosthetic arms from DECA, and this is a very complex task, actually, in which a ball is placed in space. It's elevated off of this little table, and her job is to reach out in three-dimensional space, which she can do, and then grasp the ball. So here's a couple of examples where she did well, but by and large, in the two patients that did this, you know, they were around 50%. The blue bars are how many times they actually touched the target so they could get the hand there, and then the red bars are how many times they actually navigated to the arm and then grasped the ball. And so you can see there's some challenges here. The ball moves around. This is a pretty complicated task. People are doing pretty well. However, if you look at this, this is great for people who are unable to do anything. But why can't we do a lot better? What's wrong with this? And there are really a lot of things. So a whole bunch of them are engineering, and I won't go into those. But some of them raise some very fundamental neuroscience questions. Why can't we be faster, more dexterous, and more flexible? Why can't we recreate naturalistic arm movements? So now I'm getting into the sort of what are the open questions? And I think the big open question here is that there's a big gap in what we can actually study in the nervous system. We can look across levels, and at the sort of micro scale all the way to the macro scale of looking at how the nervous system works. And I think we have some pretty spectacular tools, some of which you've already seen, to understand single neurons, their activity. We have tools to study those. We actually have more and more tools to study local networks, connectionally, functionally, look at their patterns of activity. The microelectrode array is one of those. There are others like calcium imaging, which is another promising and developing technique to look at local patterns of activity. And we can also look at global patterns of behavior in indirect measures like MEG or MRI. But we actually, the big missing level here is really looking at networks of cells, and not only just the local network, but how these networks are connected to each other, or a kind of network of networks. And so we are really unable to understand how these networks interact with each other at this meso scale. So I frame this problem as one of rules and tools. We don't have the tools to establish the rules, and we can't test the rules because we don't have the tools to test them. So if you think back of now as a more realistic view of what's going on in planning and reaching, there's really this very complex network. And in really to understand fully and to recreate these actions, we need to understand how networks compute, how they flexibly reassemble as planning goes on and action goes on. And we need a means to actually not only pick up these activities, but we also need a means to experimentally manipulate them, use some kind of stimulation. So fundamentally, the question, I think, is how does the brain function as a system? And this is something that we need to understand how to make a better BrainGate, but also if we're going to understand how plans and actions evolve, we need to be able to understand this system level activity. And both of these play into each other, as I hope is clear already. So the kinds of questions we have real trouble addressing are, what kind of properties emerge at network scale? What's different about when you have large networks than what you can see at small networks? How many neurons make up an ensemble? How many areas do you need to make up a network? And what are these computations that emerge at scale? In order to do this, I think we're going to need much better multi-area, multi-neuron sensing and stimulation techniques, and we need them with high specificity. We can address unidentified neurons. In order to make any sense out of the kind of immense amount of data that would be generated by the ability to do this, will be some kind of big data analysis, and particularly to be able to look at what we call the collective dynamics. That's a way to describe the ongoing evolving network interactions. And this is something that's beginning to be developed. There are a number of labs looking at how these state changes in networks evolve over time. I'd be glad to talk about that if you'd like. And then, of course, we need brain theory. We need to develop a sort of theory of how the brain is working. But these models need to be data-driven from these tools, and they need to be validated through data. And I think they need to be directly linked to computational models so that we can test these ideas. And these will require teams, and I think that the teams are evolving, where we need neuroengineers, neuroscientists, and especially computational neuroscience to work together to be able to solve this whole cascade of problems. So all the work that I've been doing is really part of a team like this. I'm very honored and blessed, really, to be able to work with such a great team of neuroengineers like Artur Nemiko, Michael Black, and Wilson Trukalo, and Lee Hochberg, who runs the clinical part of the trial. So to end where I began, in order to be able to make this work much better, we really need to solve the problem of how the brain actually works, but it works at a scale where we don't have the tools and we don't have the rules of operation yet. And I think that the exciting thing is that both of these two endeavors will drive each other. So thank you.